My name's Matt Flegal. I'm the Associate Director of the In Vivo Sciences group here at Charles River in Shrewsbury, Massachusetts. And I'm going to be talking today about a little bit of a clickbaity title, but it's accurate, right? It's the known unknowns. So what are the things that we're doing to bring CNS dosing in laboratory animals into maturity? So by way of introduction, we are delivering drugs into the central nervous system for a whole host of reasons. Obviously, there's a lot of focus on Alzheimer's research, dementia, whole cavalcade of things and diseases that we want to approach. But when you're looking at it from the drug development standpoint, it's tough to get a drug to cross the blood-brain barrier. The blood-brain barrier is a semi-permeable membrane. It's actually a series and system of membranes that's designed to keep things that we don't want to get to the brain from getting to the brain. Unfortunately, that also includes therapeutic drugs. Now, you can work with your chemists, and there are ways to bypass the blood-brain barrier, but when you have a molecule, it tends to take a lot of work to get it to make that transition. That then leads to the problem of, if you're going to spend thousands of hours, and if you're going to spend potentially millions or tens of millions of dollars in development on your compound, you kind of want to know if it's actually going to work first before you put all that effort in. Because all that effort that you're spending on a compound that doesn't work is effort that you could have spent on a compound that would have. You're not helping the patients if you're spending your time not bringing drugs to market. And additionally, there are actually some clinical drugs that require direct dosing into the central nervous system. So in the development phase, we're doing a lot of how do we bypass the blood-brain barrier to see if that compound is going to do what we hope it will. So there's an in vitro component, benchtop computer modeling, isolated tissues, but at some point you're going to go to in vivo testing. Simplifying this a little bit, CNS dosing falls broadly into two kind of modalities, two approaches. You've got your parenchymal approach, which is I'm putting my compound directly into a target tissue. I'm looking for a specific, motor center of the brain, a specific structure. I'm putting the material and depositing it right there. The advantages of that are, gives you a very focused target. If I only want to address the hippocampus, well, if I only inject into the hippocampus, that's all I've done. There does tend to be a little bit of leakage and distribution, but the vast majority of the test material stays where you needed it. It is a relatively limited clinical application because when it goes into humans, they don't tend to want to do brain surgery if they don't have to. There are a lot of potential complications with that. So you do that if you absolutely need to, but the tendency is, can we deliver it into a system that will carry the drug to the brain without having to make craniotomy, a defect in the skull, etc. And that's where we use cerebrospinal fluid, CSF. It's a liquid that the brain is suspended in. It acts as a shock absorber. It acts as a waste removal system. It acts as a nutrient delivery system. Has a whole host of functions. But for us, it's a liquid that we can put a test material into, a pharmaceutical compound into, that will then diffuse into it and be carried all throughout the central nervous system. So your CSF is a carrier, which means if we can gain access to it, we can use that to bathe the entire brain in our pharmaceutical compound. Now, this is difficult, right? So the central nervous system is probably the most protected organ in the entire body. The skull, the vertebrae, you have protective meninges and other tissues. Again, your body wants to keep your brain safe and make it as hard for injury to reach it. which means then that it's hard for us to dose into it. So if we're actually dosing into the brain itself, what we tend to do is use a superficial structure, in this case, the bregma, and through trial and a lot of testing, we've come up with three-dimensional coordinates that if you start with bregma as your zero coordinate, so it's your initial coordinate, and you go this many millimeters laterally, and this many millimeters rostrally or forward, this many millimeters deep, you should hit the tissue or the target site that you're trying to get to. Now, in larger species, non-human primates, dogs, etc., we do have the potential, and we use that within CRL, to do an individual MRI scan of the animal, identify in that particular animal where that target site is, and then using a computerized system, we will actually bring our dosing needle to that exact site. It's very time-consuming. It's frankly very expensive. So it's not something that you're going to do necessarily routinely. So what you're going to tend to do, actually before I get there, so let's talk about clinical experiences, right? In human medicine, there's a tremendous variety in how we get a drug into the intrathecal or the epidural space. So those are the spaces filled with cerebrospinal fluid that encapsulate the spinal cord itself, which makes it hard to derive lessons, right? Because there really isn't a consistent standard. There are literally dozens of different types of spinal needles. Certain practitioners like one, certain practitioners like another, certain practitioners don't have a preference and will use whatever is currently stocked in the room. And even the success rates are often both contradictory. So some references will say 90%, some will say 70%, which is right, hard to tell. But also they tend to be indirect. So as an example, there's a fairly recent paper that looks over the broad literature and suggests that epidural pain management has about an 85% success rate. What it doesn't answer is that because in this case, an opiate didn't make it into the right space? Was it the patient's response to it? Did it have anything to do with needle type? Did it have anything to do with duration of injection? It doesn't tell us that. So we have to figure that out for ourselves. And again, they're published papers. For those of you who have spent a lot of time reading them, especially in the in vivo space, they don't tend to get much into the way of descriptions. It'll say, we put a needle here. What angle? How deep? How long? What did you do to hold? What are the nuances? They don't really get into that. And also, if you reach out to the authors of the papers, what you'll often find is the author of the paper wasn't the person actually doing the procedure. And so it happens with a rather depressing degree of regularity that when they do talk about procedure specifics, those are often inaccurate. And if you reach out to the authors and they put you in contact with the person who is actually in the lab doing the work, they'll tell you something different that was what was in the paper. And there's great variability between the different references. So it's difficult to identify which one is one that you should follow. So let's go back to how do we find those coordinates? We use a stereotaxic atlas, in this case, the most recent version of the mouse brain. Okay. And it will give us a series of sections of the brain, and it will tell you this is how far you are from bregma, this is how deep you are from bregma. And that should, in theory, answer the questions. But it doesn't. Because if you read the introduction, what you will see is it's a pretty small sample size, right? It's 26 very specific C57 black J6 mice in a weight range of 26 to 30 grams. And we know that depending on the strain of mouse or the strain of rat or the strain of non-human primate or dog, even the average changes, right? An H-tau mouse has a wider head. Its lateral ventricle is further out. So if you don't read this particular part of the introduction, you may think, well, I can use this atlas for everything. You really can't, right? So there's a lot of, let's test, let's make sure that the site that we think that we're hitting is one that we're hitting. So again, what are the standard? What are the globally accepted coordinates? We don't have them yet. Again, this is a discipline that is finally entering maturity, but it isn't there yet because we don't have these questions answered, and those answers generally accepted. So what are our known unknowns? Okay, just talking about it. What coordinates should we use? What coordinates should we use for each different strain? Are there different coordinates for different ages? Spoiler, yes. Sex, yes. They might be very slight, but they could be impactful. We also see generational differences. So using lateral ventricle, and I'll talk about that in a moment, but it's basically a fluid-filled cavity within the brain, one on each side. So each hemisphere has a lateral ventricle. It's filled with CSF. That's part of the conduit to get into the CSF for drug distribution. If you look over time, the exact same strain of mouse in the exact same weight range, you'll see them drift out a little, come back in a little, drift back a little, drift forward a little. And that seems to be a generational thing. So even within the exact same species, strain, weight range, sex, you may actually see over time those coordinates change slightly. So again, talking about the maturity, we have tons of information. We have tons of anecdotal experiences. But are the conclusions that we drew from all of those accurate? And how are we going to test them? Right, so here's another known unknown. How much or how little test material can we put into a patient? We don't know. What infusion rate is too high? Is too slow? Is there one that gets right into the perfect balance of the two? We have anecdotal data. Do we dose by body weight or expected CSF volume? That's a big one, right? Because in humans, there's a range for CSF volume. It's between 125 to 150 milliliters of cerebrospinal fluid in an adult human. 25 milliliters out of 150 is a pretty significant difference, right? And if I'm putting a specific amount of test material into a patient and it gets diluted by the CSF, that's actually going to change the concentration of test material I'm delivering throughout the patient. And that's if I'm using volume. What about body weight? Even in humans, it's a little unclear. If I have an 80 kilo human, 160 kilo human, does the 160 kilo human have twice as much CSF? We know they don't. Do they have more CSF? We think they do. How much more? We're not really sure. Do we do same calculations for an animal, right? What are we doing to minimize variability and increase the reproducibility of these models? Because we know that when we run these studies, researchers are going to take that and they're going to make decisions that put hundreds or thousands of people into action, commit millions to tens of millions to hundreds of millions of dollars. They need to make the best decisions possible. And the more variable their data is, the less sure they are of their conclusions. And having been on that side, you know, I spent 30 years in this field so far, and 20 years of it was in the pharmaceutical side, being that person who had to propose future directions based off of fairly small data sets, right? So does needle direction matter? Where's the bevel? Logically, it would seem to make sense that it does, but which is better and which isn't? Does the needle type? There are dozens of different spinal needles. Is one better than the other? Which one should we select? Where do we inject? Doesn't matter. Does it matter if I put the test material in the lateral ventricle of the brain versus the caudal, so the rear spinal column, right? So an intrathecal injection, one of the most common ones. You inject it into the space around the spinal cord at the level of the lumbar vertebrae, so down towards your pelvis. We know that there's variability between the numbers of lumbar vertebrae, so which one are we selecting? Logically, we know the further back we go, we know that there's less space to inject into, which makes the injection more difficult. But we also know that the tissues in that area are both less prone to damage, but also less impactful if you do have a small amount of damage or there's swelling that presses down on the tissue. So which spot should we be aiming for? In every dosing area, we have the same thing, right? If you're looking at the cisterna magna, the base of the skull, so your first cervical vertebrae, the base of your skull, you open that space up. Does it matter which part of that triangle you inject in? The angle? What are appropriate success rates, right? So how do you define them? Are they based off of complications? Are they based off of gross observations? You know, nothing came back up my needle. I put my needle in place and I had CSF welling into it. Picture on the right is contrast media in a rat spine. What should it look like? How does it look different in the epidural versus the intrathecal space, right? Not well published. I'll put out there if you're doing this work, if you're in the epidural space, you're going to see it bulge out in the intervertebral areas. But there's a lot of trial and error and practice to get to there. Do we base it off of known control compounds? So ASOs, AAVs, with genetic markers, and we see how well are they distributed throughout the target tissues. How do you know if your needle's in the right place? Do you use imaging? Do you use CSF flow backup? Which sounds reasonable, isn't actually as cut and dried as it might sound. And then piggybacking off of 4 into what I suppose should be 4A, what's proficiency? So what does your training look like? How do you provide uniformity across multiple people? You know, when you're in a global organization and you have dozens of people dosing, how are you sure that every person is delivering the same results? And how do you make sure that over time, they don't, without even recognizing it, change their procedure? Because even some of those small changes may alter the results. What's your validation process? How often do you revalidate? Do you trust? Do you verify? Do you do both? So here's the underlying issue. This picture of me many years and many pounds ago. We've been doing this for a long time. But until fairly recently, these types of CNS dosing were pretty specialized, and they weren't exactly rare, but they certainly weren't common. But now, with that much greater focus into CNS diseases that are going to require test materials to be delivered directly to the central nervous system, now it's widespread. Now most major companies are looking into this. So the number of studies has expanded massively. So now is the time to take it from a cottage industry and turn it into a mature one where we have these answers. Which raises the question, so what do we do now? And what I'm going to share with you is an approach that we're using within Global Charles River. Is probably applicable to most companies, right? So it's not special to us, but we're starting to figure out how to do this. And that's what I really wanted to share with everybody. So the key is it's got to be evidence-based. It's got to be driven by science and data. As a surgeon for 30 years, I can tell you that I have had many completely logical assumptions be wrong, right? Or things that I was taught that we've just evolved as a discipline since then. We do our best with the information we have, but if you're going to do this, I would propose to you, it can't be based off of anecdote. It can't be based off of conventional wisdom. It can't be based off of, I'm sure I'm right. Because when you're making decisions about compounds that are going to go into humans, that's a pretty high bar that we need to make sure that we cross. So we're doing this based off of, to coin a phrase, show me the money. I want to see the evidence. I want to see real data. I want to see numbers. And if we have numbers, then that should be able to drive actual harmonization. And so that's what we've committed to. We're going to do this stuff at its core the way that we've identified as best practices, again, driven by actual science, actual analysis. That means looking back. The good news is we've got a data set at our sites. We've been doing this for 30 years. So what does the data say 25 years ago, 10 years ago? What can we learn from it? And then how do we design studies around our actual success rates? Is it 90%? Okay. Is it 95%? Great. How many subjects do we need per group to give actual robust data? What's the variability within the dosing? What's the variability within the molecule types, right? Because if we're going to do this, if we're going to use lab animals and make sure that we're not wasting their sacrifice, right? That we're not contributing poor data or potentially misleading data. We need to understand that stuff. So we're digging into it. We're using those different molecules and seeing if there are differences. So here's what we've done, and this is what I would suggest. This is what works as a template. You got to get a holistic group put together. So we've pulled in our surgeons, our dosers, our technicians, our scientists, our prosectors, our pathologists, our study directors. And we're having them all work together, not share information back and forth. I'm saying in a room together, and I'm going to touch on that in a minute. But I cannot tell you how important this is, right? If this stuff is siloed, I can tell you it will not work. I've tried at multiple companies before. It doesn't work because you miss things, right? So it comes back to put the people doing the work in the same room, showing each other what they're doing, walking through the differences, sharing their anecdotes, sharing what's worked, sharing their experiences. You will get more accomplished in 2 days than you will get accomplished in a year. And that is actual experience talking, right? Because people walk out of that room when they've actually worked together and seen what's going on and shared the nuances and asked the questions while things are actually happening. They walk out of there confident in each other's abilities and what they're bringing to the table. Because at the ultimate end of this, you want to be a partner, right? You want to design the best studies to get the best results. So again, the holistic approach. Intrathecal, where do we go? Well, the higher we go, more space there is. However, the higher you go, the more risk there is of damage in neurologic signs. So logically, as a surgeon, what I've always thought is I want to go further towards the pelvis. But if you talk with your prosectors, they will tell you getting a good tissue sample out of L6, L7, or even L5, L6 is really difficult, which means lots of times your tissue isn't of the quality you want, which means your analysis isn't of the quality you want, which means your results aren't of the quality that you want. So when you put all of us together, we go, oh, you know what? Let's find a happy medium. But had we not talked to the people who were actually taking the tissues, never would have known that. Never would have occurred to us to ask. But because they're at the table putting out their own experience, you get those answers. Look, I'm a surgeon. We're tough to get, right, to any kind of consensus. What's the joke? You put 5 surgeons in a room, you get 20 different opinions. You put 1 surgeon in a room, you'll get 3. But if you put them in a room together and you let them share and you let them talk to each other and see each other work, you're not hurting them. They're coming to their own consensus. And I will tell you again, 5 minutes of actually watching someone do a thing is so much more useful than even 5 hours of listening to them try and describe it, because it's never the same. You never get a feel for the nuances. And frankly, if you're passionate about what you do, you want to see it work. Everyone's bringing their assumptions to the table, and you got to challenge them, right? So much of what we do is based off of limited data sets. So we're building the data sets and pulling all of our historic data so that we can actually see what is happening, and having those multiple points of view, not just to poke holes, but to say, what about this? Invaluable. Because we tend to think of harmonization as a dirty word. And if you do it wrong, it probably is, right? Because if you get a harmonized process that's put to you, well, if you're passionate about what you want to do and you're committed to high quality, well, you're being told to do something that is different than what you feel is the best way to go. So you resist, not because you're arrogant and not because you're inflexible, but because you're committed to delivering the highest quality results. And this may not track with it. But when the people doing the work drive the harmonization, you're not telling them to do a thing. They're coming out and telling everyone else the best way to do it. You've got buy-in because they were part of making the process in the first place. I would propose to you that's how harmonization should work, right? It's the people who have the most skin in the game driving what those harmonized procedures are. There's a global monitoring approach. So are you assessing every study? How did it go? What worked? What didn't work? Can you pick up early trends? Are there new best practices developing that you need to put into some sort of firm description and then train everyone on the new, better way to do things, right? Because a best practice is great until you identify a better one. And how do you not lose those better ones? Because again, that's how you drift over time, is people finding a way that's better than the standard and with some justification saying, I'm going to do this better way, but it hasn't been shared so that everyone can benefit. Again, what are success rates, performance, sites, methods, personnel? I don't know. Maybe I'm the worst doser. We should know that, right? And that's what we've really dug into, is how do we identify that? Because if I'm trying to do the best job I can, I need that feedback. So if I'm doing something wrong, that can get identified and repaired. Again, the best practices. And then also, as this evolves, what do the people that we're generating the data for? And we're looking externally, but having run these labs internally, what do your development groups need? Are you sure that your process is going to deliver what they need? Are you talking with them about what their specific requirements are? I need this tissue. I need to look here, right? It's got to be a partnership and a discussion, because you don't want to build a thing that's not useful to the people that you built it for. And then finally, how do you standardly train, right? And that's hard. How do you develop a curriculum? How do you minimize the differences, and how do you use novel technologies, right? So we're working on 3D scanned models, because if I'm training a new surgeon, if I can have a mannequin that they can train on for 2 hours every day, not using any animals, so there's no wasting animal lives, they can focus on the key parts of the procedure. And I would propose to you that if someone is learning and they've practiced dosing 2,000 times, they will be better than someone who has done it 20 times, right? What's the training software? What are the various, modalities that we can use? Virtual reality. There's actually surgical virtual reality training. So how do we bring all of those things into play to give the people doing the work the most support? All right. And that's my last slide. So I appreciate you all, hopefully, not tuning out and going to do something else for the last 30 minutes or so. If you have any questions, there is a submission. I will be happy to answer them. Again, anything I can do to help people from avoiding reinventing the wheel, we've all done it. It's not fun. So helping all of us out, I think, is a really good thing. So thank you.